Hey guys! So I had a friend ask a question, and the easiest way to answer is make a video, and I'll probably end up sharing it with everybody. So the question was, what are the enzymes at the end of DNA transcription? So after DNA has been transcribed into mRNA, and mRNA is starting to go off, and it's going to do its own little thing and become, a protein eventually. Before it goes out of the nucleus, the mRNA has to be messed with to make sure that it looks just right. And there are certain enzymes that do this. There's also enzymes that make sure we don't have mRNA stick around too long. So what we're talking about in this video is what enzymes mess with mRNA and how do they do it. So there are three. There's C, and then SN RNA. So miRNA, siRNA, and snRNA. C, and then SN. C, SN. So the Mi and the C, we'll start with those. miRNA, is a general sort of mRNA repressor, and so it tags it to slow it down, and it will add tags onto it, and it's sort of a general repressor. And whereas siRNA is very specific to the RNA it's after, the mRNA that it's after, and instead of generally tagging a bunch of things, it will go to one and it will tag that one. And when things get enough tags, then they're going to go to the proteasome, is where I think these ones go, and they will be chewed up and they'll go away. So both Mi and C play a role in inhibiting and in the eventual destruction of mRNA. And so that's why we don't have mRNA floating around forever or until it falls apart, because otherwise you would make like, I have 10 copies of make protein X, and whenever they bump into a ribosome, we'll make protein X until they magically fall apart. So we'll make protein X for a very, very, very long time, and we don't need that much. I guess it's government work, right? So anyway, so that's what those two do. The last one is snRNA, and he has the longest story. So mRNA comes out, and it's got, actually, we're really clever, or whoever designed us is really clever, in how we got multiple possible proteins in one string of DNA, okay? And so we make an mRNA out of that DNA sequence, and depending on which bits we remove and which bits of the sequence we leave in, we'll get different proteins. So one sequence can code for a lot of different proteins based on which little bits we take out. And if we take out a bit that's supposed to code for a proline, proline is an amino acid that makes a bend really, really well. And that bend would make so the protein folds differently. And so if we just take out that one bend, then we get a whole other protein in the primary and secondary structure of protein folding. And so the way that we take advantage of this sequence being able to make multiple proteins is we take snRNA, who will come in and take out whatever bits, and we call these introns. It'll take out the introns of a specific protein sequence. The snRNA knows which ones we want to take out for a given protein. We don't know how it knows that, or at least I don't. I haven't read anything where we know. And everything that's left in this mRNA is called an exon. So with all the introns taken out and the exons left in, the only thing that ends the snRNA's major job. It may take, after that, I don't remember if it's snRNA. I think it probably is. But the mRNA, once it is finished being altered, it will have a cap and a poly A. So it's just going to be like the nucleotide AAAA, adenosine, 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 all just along the end. So there's a cap and poly A tail. The cap is on one end, the poly A tail is on the other, and then it'll be sent out of the nucleus. The cap, once it gets out of the nucleus, is what is going to allow it to hook to the ribosome properly, and the tail will, both the cap and the tail, will prevent it from degrading because of bumping into things or whatever, because you wouldn't want it to have bad luck and be broken off at the end, or there are charges, is actually what it really is. There are charges on, if you don't have it capped, then there's a charge, and that charge could stick to things or have things come and take it and tear it off, is my understanding at least. Definitely it's for stability. And yeah, so cap, poly A tail, introns out, send out the mRNA, meets with the ribosome, does its thing, eventually me and C RNA will come and chew it up. And by the way, it'll probably be a test question for this last bit. Notice that the me, C, and snRNAs, they're RNAs. They don't make a protein. They are active and doing things with no protein. And that really wigged out a lot of people's minds when we first found that out. And so it's probably going to be a test question. Do you have, enzymes? Are all enzymes proteins? No. Some of them are RNA. And ribosomes, those are RNA. There's a little bit of protein in them, but the active parts of them are RNA. And so we see that not all enzymes or active functioning machines in the cell are protein, just most of them. So hopefully this helps, and yeah. Have a good one.